Hello, and welcome to this Nature Research custom webcast titled Enhancing Immunotherapy Against Solid Tumors. My name is Jojan Karpen, and I will be your moderator. Today's webcast is produced by Perkin Elmer. We'll begin the webcast with a presentation from Eva Morschler of Bellicum Pharmaceuticals and then end with a Q&A session. To ask a question, just type it in where it says type your question here and then press submit at any point during the webcast and we'll answer them today. And now over to Eva.
Thank you for the introduction and the invitation to talk to you today about our technology and how in vivo imaging is advancing our discoveries. I will introduce you to our molecular switches and how they enhance immune cell therapy against solid tumors. My introductory slide shows the current general workflow of cell therapies. Leukocytes are collected typically from the patient that is called autologous treatment, or in some cases from another healthy donor that is called allogeneic treatment. These cells are activated, then genetically modified to express a chimeric antigen receptor, a CAR, that targets a certain antigen, mostly using viral transduction, but transposon, CRISPR, or TALENs are also used. Afterwards, they are expanded, then cryopreserved and submitted to release before infusion into patients. Up to this point, everything is highly controlled under GMP standards to know exactly what dose is being administered to each patient and making sure cells are not contaminated. Their effectiveness is tested in vitro in culture. But once the cells are administered to the patient, the physician has little control over these cells. In leukemia and lymphomas, where the tumor cells target antigen is accessible, tumor killing efficacy is high, but so is the incidence of side effects like cytokine release syndrome, graft versus disease, and on-target off-tumor attack like the T-cell elimination in the case of CD19 CAR-T therapy. When the target is on a solid tumor, the T cells have to home to the site, enter the tumor, and recognize the target antigen. T cell activation also requires co-stimulation from antigen-present cells that may not be present. The tumor microenvironment is usually high-sick, has immunosuppressive cells and mediators like TGF-beta, inhibiting T cell function and proliferation that is necessary for sustained tumor killing. The checkpoint molecules that are regulated on tumor cells, other cells in the tumor, also inhibit T cells. That is why checkpoint inhibitors are successful, since they remove the brakes on anti-tumor immune response. Bellicum's approach has been to develop molecular switches that are transduced together with the CAR cells, and they are silent until a small molecule is present, binds to the binding domain, and initiates cell signaling domain. Based on the signaling domain, can either promote survival and expansion or induce apoptosis to reduce T cell numbers, leading to our goal of an improved benefit ratio. First, let me introduce to you our activation switch. On one side of the slide, there is a schematic of the endogenous T cell receptor, T-cell complex, and the CAR molecule was activated by a target antigen and signaled through the zeta chain of the CD3 molecule, which activates the N-fast transcript factor and results in interleukin-2 cytokine production, the sure sign of T-cell activation. On the other side of the slide is the schematic of our activation switch. The binding domains are mutated variants FKBP and immunophilin, and the small molecule that binds to it with high specificity and affinity is called rimidacid, or RIM. The signaling domain consists of parts of MyD88, that's myeloid differentiation factor 88, and 40, parts of CD40. And it is inducible. That is why we call it IMC. Upon binding of remedies, the FKBP domains arise and activate the signaling domains, leading to activation of the NF-kappa beta transcription factor that promotes cell survival cytokine production. There are other transcription factors that are also activated, and more information about this can be found in a paper reference on the bottom of the slide. To show effectiveness of our activation switch technology, we used a subcutaneous HPEG xenograft tumor model in NSG mice. HPEG is a human pancreatic cancer cell line with high endogenous prostate cell antigen, PSCA, expression that we can target with our IMC PSCA CAR T cells. The tumor cells were transduced with firefly arrays to enable monitoring of tumor growth by in vivo imaging, besides caliper measurement. NSG mice are immune deficient. We purchased them from the Jackson laboratory, and they enable us to use tumor cells and T cells without rejection, although graft versus host disease can and will still develop with time. The T cells originate from healthy or PBMCs. The tumor cells in the cells are not matched, and that is why we use control T cells without CAR or the activation switch to see the endogenous activity of the T cells against the tumor. The images on the slide show 3 groups of mice. They all received tumor cells subcutaneously, and 7 days later they received T cells injected through the tail vein. There's the control group, and then 2 groups that received IMC PSCA CAR T cells. One of these groups received weekly IP intraperitoneal injections of rimidacid, while the other one received vehicle. In vivo imaging was performed every week starting before the T cell injections. The imaging itself is performed under anesthesia. The mice acted intraperitoneally with D-luciferin and 10 minutes later imaged in the iris imager that takes a photograph and a bioluminescent image and overlays the two so we can see which parts of the body the bioluminescent signal comes from. We can adjust the scale of the artificially image to show the amount signal based on average radiance that is calculated in the living image software. The more orange-reddish color you see, the higher the signal. Having the images next to each other gives a simple and powerful way to show changes in tumor growth. See that the control group could not control the tumor, and the mice had to be euthanized 28 days T-cell injection. The IMC PSCA CAR T cells that were treated with vehicle in the middle controlled the tumor better than the control T cells, the CAR, but the last group that received treatment had much better tumor control, and at day 28, there was only one tumor visible that disappeared by the end of the experiment. On this slide, we can see the results of tumor monitoring with in vivo imaging and caliper measurements side by side. The top panel shows the tumor area measured by caliper where the limit was 100 square millimeter. To calculate the tumor area, we measure the length and the width of the tumor, which is quite objective. The tumor growth was homogeneous in the control group, but not in the CAR T cell treated group that received vivo. The dotted lines show data from individual mice. The straight bold line is the average. some mice showed tumor control while others followed the path of the control. In the CAR T-cell plebiscite group, tumor control is achieved in all the mice. The lower shows the average radiance from the bioluminescent signal on a logarithmic scale, and it's very high both in the control and the treated group due to the high signal in some of the mice. In the immunotherapy treated group, we see a several log drop in the signal, even though there's more difference between individual data here at the lower level. The caliper data, shown on the top, is better at resolving bigger tumors. Imaging data that we saw on the bottom is better at resolving differences and showing smaller tumors. This slide shows the results of T-cell monitoring. In a separate experiment at day 21, blood was collected and tissues were tested to identify CAR T-cells using quantitative PCR-based vector copy number assay. Taqman primers and probes were designed to recognize the signaling domain, and plasmid DNA was used for standard. Genetic signature of CAR cells were found in the blood, tumor, and the organs examined in the mice that received immunotherapy treatment, while none of the vehicle-treated groups showed any sign of the T-cells at day 21. This result shows that the activation switch supports T-cell survival. And when you have activated T-cells all over the body, you expect to see some side effects and would like to have a tool to remove the cells if this having a safety switch is advantageous. As you can see on the schematic on this slide, the binding domain in this switch consists of a combination of an FKBP and an FRB domain, and the small molecule that links these heterodimers is rapamycin. Rapamycin is an immunosuppressive molecule first found in bacteria, and it binds wild-type FKBP12 and FRB with very high affinity. The signaling domain in this switch is caspase 9, which is an initiating component of the apoptosis pathway, and it is activated by dimerization. On the other side of the slide, you can see images generated by in vivo imaging. NSG mice were infected with human T-cells that were transferred to the safety switch and fire proliferates. One group of mice with IP injections of vehicle, 3 groups received increasing doses of rapamycin, and 1 group received remedies. Mice were imaged right after T-cell injection, 1 day later before treatment, and then 5 hours and 24 hours after treatment. After tail vein injection, the T-cell signal is visible in the area of the lungs. A day later, the signal is somewhat reduced and diffused. A single rapamycin injection results in a significant drop of T-cell signal even at 5 hours, and at 24 hours, the signal is completely gone. The signal is still visible at all points in the vehicle-treated mice and immunotherapy-treated mice, showing that the low binding rate of immunotherapy to wild-type FKBP provides adequate specificity. We have performed further studies and could not reach a high enough concentration of immunotherapy in vivo that would activate this safety switch. However, it is achievable in vitro in cell culture. This shows on one side the bioluminescent signal, cells measured at 24 hours, the drug treatment, and next to it are the results from flow cytometry analysis formed on spleen homogenates, also 24 hours after treatment. The safety switch contains an epitope from the human CD19 protein that we can use as a marker. Our genetically modified can be identified as negative mouse CD45 and positive for both human CD3 and CD19. The result of the analysis is identical to the result of the in vivo imaging, and this saves us time, materials, and effort that would be required to harvest spleens at every time point to analyze T-cell biodistribution, either by the vector copy number assay or by flow analysis. Using in vivo imaging, we get real-time data faster. As I had mentioned, we proved that there is no crosstalk between our switches due to the binding specificity and affinity of the small molecules rimidusine and rapamycin. So we felt confident to put both switches into a CAR T cell, as you can see in the schematic on this slide. We call it dual switch technology. The antigen-specific CAR is the main player that provides tumor recognition and killing. Our IMC activation switch that is turned by rimidusine administration promotes survival and provides co-stimulation for the CAR. Then we have our safety switch that can be activated if needed by rapamycin administration, leading to apoptosis and elimination of activated T cells. This slide shows our dual switch technology in a solid tumor model using HER2-expressing OE19 tumor cells and our HER2-specific dual switch CAR T cells in NSG mice. We wanted to see T cell expansion in real time in parallel to tumor growth, and it wouldn't have worked with firefly luciferase in cell types. Perkin-Elmer introduced a new substrate called colanterosine that works with ranilla luciferase and allows us to image the mice and follow the T cells in real time. We can't introduce both substrates at the same time since the readout is bioluminescent light, not different based on how it was generated. Simply put, the inputs are different, but the output is the same. So we image the microtumor one day and a few days later for T cells to give them a little rest, able to clear the substrate and the effects of anesthesia. The tumor is located on the back of the animal since we inject the cells there under the skin, while the T cells are distributed all over the body. When we image for T cells, we take an image on the dorsal and another on the ventral side. On this slide, we show the image tumor on one side of the slide and the image on the ventral side of the animal, the T cells next to it. On the ventral side of the body, the T cells show up in the spleen and in the digestive tract. That is one of the biggest immune of the body with lots of lymph nodes. We had 3 groups of mice in this segment. The control group had T cells that didn't contain the CAR, only the safety and the ranilla luciferase. The other 2 groups contained dual switch CAR T cells, and one group was treated with vehicle and the other group was treated with rimidusine to trigger the activate switch. By day 17 after treatment, the group that had a boost from the activation switch had almost no visible tumor, while the vehicle groups still had tumors, although they were smaller than the ones in the control group. The next 2 weeks, several had to be euthanized due to large tumors in the control and the vehicle treated groups, while the rimidusine treated groups had no tumors. If you look at the T cell signal, it is a little bit misleading that there doesn't seem to be any signal at the beginning of the experiment, but that is because of the scale we use. The T cell signal is so high at the end of the experiment that we had to increase the scale, otherwise these photos would look like they were overexposed. You will see on the next 2 slides quantification of the signal, and hopefully it will convince you that rimidusine-induced stimulation significantly increases T cell signal. Also, at day 25, there's a mouse that doesn't seem to have any T-cell signal and is marked with a little white star. That is actually an injection error that was not noticed and corrected at the. On this slide, we show the data from tumor monitoring. On the side, we show the data from in vivo imaging. On the other, we show the tumor volume, which is a result of caliper measurement. This tumor type grows tall, while others are flat. So we added a measurement and calculated tumor volume instead of area. Data from the control group is indicated as a black line. The vehicle-treated CAR group is shown in red, and the Rimidacid-treated CAR group is blue. The bioluminescent signal and the tumor volume data indicate that while the dual-switch CAR T cells have some tumor-killing effect by themselves, when the activation switch is turned on by the administration, there is a synergistic effect and the tumors are easily eliminated. On this slide, we show the bioluminescent signal from the T cells in the ventral side, and you can see that the signal is very low at the beginning and stays low for the control and vehicle-treated groups, but it is several logs higher in the Rimidacid-treated group. There is significant T-cell proliferation in response to tumor plus activation switch signal. To prove that the CAR T cells are highly activated, we took blood samples 19 days after T-cell injection and measured cytokine and chemokine levels from plasma using a multiplex assay. The data is presented as a heat and fold changes are indicated in the color scheme, where red shows upregulation and white is downregulation compared to baseline before T-cell injection. In the control group, none of the mediators increased. The vehicle-treated CAR T-cell group had some slight increases in several cytokines, but the group where the activation was turned on by Rimidacid, we found several cytokines and chemokines that were highly increased. It has been observed, we saw it as well, that activated CAR T cells produce a mixture of so-called T helper 1 and T helper 2 type cytokines, like interferon gamma and interleukins 5 and 13. The most highly upregulated cytokine that we found was interferon gamma. It has a wide range of effects that anti-tumor immunity. The next highly upregulated indicator was granulocyte monocyte coagulating factor, GM-CSF, which is a hematopoietic growth factor and immune regulator, and has been used as adjuvant to increase dendritic cell maturation. These cytokines can recruit the immune cells of the host to the tumor to aid the CAR cells in eliminating the tumor. We can't model this in an NSG mouse since that mouse is immune, but we are working with syngeneic models using immune-competent mice to show this effect. TNF-alpha and IL-6 are also produced by these CAR T cells. These cytokines have been implicated in adverse side effects like cytokine release storm and neurotoxicity. We have the safety switch technology enabled in our dual switch CAR T cells to be able to curb the cells if adverse effects are observed. This next slide shows the in vivo imaging of dual switch CAR T cells that were pushed by high tumor burden and increased co-stimulation activation switch. The images show T-cell signal in all the treated groups and almost none in the vehicle group. At day 15 after T-cell injection, temsirolimus was administered IP at increasing concentrations. Temsirolimus is a derivative of rapamycin as a cancer treatment in the clinic and works equally well at turning on our safety switch. A day later, the T-cell signal was reduced and stayed low till we restarted immunotherapy treatment to see if we can reactivate the remaining T-cells. I would like to point it out that by this time the tumor was eliminated in these mice, so no antigen driving T-cell survival or proliferation, except in the control group where the tumor is still present, but there's barely any T-cell signal. In the remidosib-treated groups, the remaining CAR T-cells were able to expand again. This slide shows the decrease in bioluminescent signal after activation of the safety switch, demonstrating a nice dose-dependent effect. As I have shown you earlier, these activated T-cells release high amounts of interferon gamma and other cytokines. The graph shows the plasma concentration of interferon gamma before, these are the black dots, and a day after the activation of the safety switch, the red dots. There is a dose-dependent decrease in interferon gamma concentrations that correlates well with the decrease in bioluminescent signal. We found similar dose-dependent decreases in all the other cytokines and chemokines that were elevated before. We believe that rapid removal of highly activated T-cells and reduction of levels of inflammatory cytokines can stop adverse effects and allow the body to recover. On the other hand, we don't want to eliminate all our CAR T-cells in case there's a regrowth of tumor. We have shown that the remaining dual CAR T-cells can be reactivated and expanded by turning the activation switch. Please allow me to quickly summarize the main points of my talk. Wellicome's activation switch technology enhances CAR-mediated tumor killing via increased T-cell proliferation and cytokine chemokine release. These mediators provide stimulation for the cells of the host to further improve tumor control. Our safety switch technology provides dose-dependent elimination of activated T-cells and reduction of immune stimulators to reduce adverse effects. The dual switch technology enables us to target tumor antigens that are also expressed on healthy tissue at lower levels, like PSA or HER2, because if the CAR T-cells attack healthy tissue, we can reduce their number by turning on the safety switch. It is all about balancing activation and reducing side effects. Last but not least, I have shown you that tumor growth and proliferation can be monitored simultaneously in real time with vivo imaging using different bioluminescent substrates as a powerful demonstration of CAR T-cell efficacy. This work was performed by a group of excellent scientists at Wellicome, and I am proud to be part of the research and development team. I would like to thank Drs. Dale, Foster, and Duong as answer for their guidance and leadership, Kelly, Mary, and Matt for all their work. I would also like to thank several people at Elmer, Julie, Will, Janet, and Alexandra for support and continued collaboration with us made this work possible. Thank you all for your time, and I'm happy to take questions.